Reshma, you can proceed, ma, please.
Okay, sir. Thank you, sir. Good evening to all. I hearty welcome you all to day 4 of our faculty development program. On behalf of our Sacred Heart College, Tirupati, it's my privilege to welcome you all to this 5-day online faculty program, Global Scenario and Future Prospects in Life Sciences, organized by PG and Research Department of Biochemistry. It's my pleasure to welcome the esteemed resource person, Dr. M. Kumar, Assistant Professor, Department of Botany, Madras Christian College, Autonomous, Chennai. I heartily welcome you, sir. Thank you, sir. Thank you for accepting our invitation and great support to being with us to share your knowledge. With my immense pleasure, I would like to welcome Dr. Jayaprakash, sir, PG and PhD head, and Dr. M. Fernandez Suresh, sir, UG head for this
I will
meeting.
also
Welcome, sir. I extend my warm welcome to Dr. K. Sriram, PG Associate President, and my dear colleagues. Welcome you all. And I also welcome faculty members and a research scholar from our institution and other institutions. Welcome you all. Now I would like to call Dr. V. Gopalakrishnan, sir, Assistant Professor, Department of Biochemistry, to introduce our resource person. Please, sir.
Okay, good evening to everyone. It gives me an immense pleasure to introduce the resource person of the day, Dr. M. Kumar, Assistant Professor, Department of Botany, Madras Christian College. Sir did his doctoral research in Center for Advanced Studies in Botany, University of Madras, during the year 2005 to 2010. Actually, he carried out number of research activities in the various fields related to mushroom diversity, molecular systematics, mushroom metabolites, mycoforest prospecting, ethnomycology, mushroom food and nutrition, and mycotechnology. So far, he was guided 23 PG students and 4 MPhil scholars. Currently, he is guiding 3 PhD scholars. In the year 2005 to 7, Sir was selected as a project fellow in UGC major research projects. In the year 2007 to 9, Sir received junior research fellowship, followed by in the year 2009 to 11, he was selected as a senior research fellow under All India Coordinated Project on Taxonomy, especially in the field of fungi. It was founded by Ministry of Environment and Forests. Dr. M. Kumar, he published 3 new species and over 10 new records of fungal species from India and deposited 20 nucleotide sequences of various fungal species in NCBI GenBank database, USA. He served as a member in various professional bodies and also he received Dr. APJ Abdul Kalam Young Scientist Award from Marina Labs. Dr. M. Kumar also acted as a domain expert and reviewer for the state government higher secondary botany curriculum revision and textbook of Tamil Nadu government. Sir published nearly 25 articles in various esteemed peer-reviewed journals with high impact factors. And also he acted as a resource person in more than 35 academic events. So far, he completed 5 student projects from TNSCST. With this brief introduction, once again, I welcome Dr. M. Kumar, Assistant Professor of Madras Christian College. Now, I hand over the session to Dr. M. Kumar to elaborate its topic, microbes that changed the world. Kindly take over the session, sir. Thank you.
So thank you for the nice introduction.
Okay, thank you, sir. You can proceed, sir.
Yes. Dear organizers, can you allow me to share the screen?
Yes, sir. Yes, sir. Now you can.
Hope you can see my slides.
Yes, sir. It is visible, sir.
Yes, sir. Yes, sir.
A very good evening to one and all present here. And I am happy to be a part of this program. And I can see some of the known people in this forum. And without taking much time, I'm just going into the program. Microbes that changed the world. Yes, microbes has changed the entire scenario of this earth. Is my slide changing?
Yes, sir.
Yes.
Yes, sir. Kindly make it as a full screen. Kindly make it as a full screen. Yeah, now it's fine. Thank you.
Sir, yes, sir.
Yeah, this is the Earth 2.5 billion years ago. This is how the Earth was. Just stones, rocks, and sand particles. Although Earth was formed 4.5 billion years ago, the Earth remained a place where it cannot support any life forms. And probably this could be the Earth if we travel 4.5 billion years ago. At least, the Earth was having an anaerobic environment, and it was not suitable for anyone to live on this Earth, any life forms to live on the Earth. And 2.4 billion years ago, the Earth undergone a great oxidation event. And because of this oxidation event, slowly the Earth started, oxygenating, and then slowly one after the other life forms came into existence. And one such life form which came early on this Earth is photosynthetic bacteria. Okay. These photosynthetic bacteria are, the cyanobacteria, like, bacteria which oxygenated the entire Earth and changed the entire fate of this Earth, which was just a barren, land. Okay, so all this event happened, after 2 billion years ago. The Earth was 2 billion years after the Earth was formed, and these are the bacteria which had a common ancestor, that is the cyanobacterial ancestor, from where one of the photosynthetic bacteria, we call it as oxyphotosynthetic bacteria. These bacteria are even now, they were identified from few regions and recently reported, and they form a lineage between this group. And these are the group of bacteria or the organism which helped the Earth to oxygenate and formation of all the other life forms. 300 million years, a big crash happened on this Earth, and this could be the Earth, the older Earth, and this is the present Earth which you see. We can see a marked difference between the original Earth and the present Earth. The present Earth is so beautiful because the microbes which formed on this Earth earlier 2.5 billion years ago, they are the one which were responsible for painting the Earth with a green paint. Now you see Earth in this form. This green and different colors are due to the bacteria which originally painted the sandy Earth into this one. And we don't realize all this happened due to a group of microbes, whether it may be bacteria or anything else, it is the microbe. And all this event happened by by a process which has happened on this Earth, and we call this event as endosymbiosis. And because of that, the earth got oxygenated, and the present earth is like this. And this is how the symbiosis went. Earlier, there was a ancestral prokaryote which had only the cytoplasm and some DNA particles in it. And then later on, some infoldings has taken place in the plasma membrane, and then due to which the continuous infoldings and other things, endoplasmic reticulum formed, and then the establishment of nuclear envelope has taken place. And during this course, the prokaryotic cell, which is becoming mature, which is becoming an eukaryotic cell, they have engulfed an aerobic heterotrophic prokaryote, and that became the mitochondria, and that formed separate lineages. And then during the same course of evolutionary process, those prokaryotic cell which had engulfed mitochondrion like prokaryote has also engulfed another photosynthetic prokaryote. We later call that as a chloroplast. Okay, so they got engulfed, and that is how slowly a prokaryotic system became eukaryote. So all the life forms were dependent upon one single the prokaryotic microorganism. Okay, so and because of this microorganism, the eukaryotes formed, and that became the marked change on the development of different life forms. This picture also explains how the modern photosynthetic eukaryotic form and the modern heterotrophic eukaryotic form. And when this prokaryotic microorganism was, if that is not existing on this earth, or not formed on this earth, or not being engulfed on this earth, probably we would not have come across the eukaryotic organism, which which formed several other advancement during the evolutionary process, and the higher human beings are formed at the topmost of the evolutionary process. And for all this event to take place, the mitochondria as like a microbe, or the chloroplast like microbe was the one which was responsible. But many times we don't feel that it is the microbe which formed the basis for the developments happened on this earth. But then this is true, and these engulfment has beautified this earth, not only the globe picture, and in realistic also the earth is like this. And we also go to green places where we refresh our mind. So all this is due to the microbe on this earth. It is not the plant which is green in color. When we see a plant is in green color, it is not the plant, it is the microbe that gives green color. Not a single green. You can see different shades of green. So all this are not plant, not because of plant. It is the one which is hiding inside the plant, which gives the greenery. So we always admire plant and its green color, but we have to understand the fact it is not the plant which is green in color, it is the microbe which is green in color. Okay, and that is the one which is hidden and reflecting to our eyes. And not only that, the microbes have done several changes, or they have marked so many things on this earth, and it's change. What we see today is because of the microbe. Another important event which has taken place on this earth, according to Vedas. Okay, there was a bana called Soma Bana, drink. We call it as a drink. There was a drink called as Soma Banam. There are several stories behind this Soma Banam. One story behind the Soma Banam is the Asuras and the Devas. They had a typical fight between both for a Banam, which is divine, divine thing which gives immortality. And for this, the Devas and Asuras were fighting. And in a writing by Gordon Wasson, is a very good researcher and a writer who has done several research, anthropological research. So he found a particular species called as Amanita muscaria, which is a fungi. We call it as macrofungi. And he identified from a particular group of people that this is the particular microbe which was added in the Somabana along with the other plant. So it is Somabana is mixture of several medicinal plant herbs, we call it as, and this is one component of that Somabana which gives immortality. And this mushroom is still called as the iconic mushroom, or wherever mushroom is told, this is the representative species, because this is called as elixir of life. And Gordon Wasson writes that this is the major constituent of that Somabana. And another literature says it is the Devas and Asuras, they were taking the Somabana from sea. So there are different stories exist, but then the main ingredient is the microbe called as fungi, which plays an important role in Somabana, which has changed the Devaloka and also the earth, which belongs to the human beings. So in such way, the microbe can influence the earth we see and also the earth we don't see. So to that extent, they are a powerful organism that can bring change. And another important change in the modern era we have emphasized is during World War. We have seen there are so many soldiers who were dying because of influenza, cholera, and many other diseases. The death rate was 18% in the World War I, and suddenly in the World War II, the death rate became 1%. So this is a great change which has been achieved from 18% to 1%. It is not an easier thing. Earlier we were depending upon people that is local medical practitioner, or the local people who do some local treatments. That is how we were dependent. But during World War, the one microbe which has changed the world through Dr. Alexander Fleming, that is in 1928. He was telling in one of his statements, I did not plan to revolutionize all medicine by discovering world's first antibiotic, but I did exactly the revolution what was wanted. This happened not because because of a big life forms on this earth. This happened because of one small mold called as penicillium, and that too a species called as Penicillium notatum. Okay, and because of we all know the story behind this, and he was finding out an antibiotic, and that yielded around 400 million units of pure penicillin. Okay, in 1942. And later on, there were so many others who started working on this penicillium. They came out with different species and different strains, which yielded much more than what was produced in the beginning, that is 400 million units of penicillin. And from a species called as Penicillium chrysogenum, they were able to yield more than 400 units, that is 200 units extra, that is 600 units. And this formed the beginning of the cure. Earlier there were not much cure, but then after this marked event which happened on this earth due to one single mold, there were so many antibiotics which were found, not only penicillin, There were so many other, antibiotics were found later on, and this one formed the beginning for all the antibiotics which are, found on this earth in the later days. And this is the mold. It looks very small in the plate. Probably, if it is not in the plate, it is growing on the substratum. We cannot even, find out what exactly it is. Such a small microorganism, but then this was able to mark a great beginning on this earth. So that is how, the other antibiotics were formed later on, and people started understanding, so many aspects of, the disease and disease causing agent, followed by this, and human beings got a great deliverance from all the diseases they had. And then later on, there were several events happened, and Louis Pasteur, during 1850s and 60s, he was able to invent, the storing of the milk by a bacteria he has identified. And then parallelly, all the other findings were, able to happen. That is, earlier people thought, the fermentation or, transformation of sugar to alcohol is through a chemical reaction. But later on, Louis Pasteur's identification made a great beginning. And then it was, found out that a microbe or a bacteria which is responsible for, the big change on this the chemical transformation. And then this marked, a great beginning in the industrial production of many microbial products. And starting from here onwards, and the invention of the antibiotic onwards, there were so many findings. Industrial revolution with related to biology, and now we call it as biotechnology, has taken place. And several products came into existence. That is how the vitamins, that is how, the minerals are now being isolated with the help of fermentation process and the other agent. Not only that, you can see the vaccines are coming up, and all this are, beginning of these findings. And Edward Jenner was able to find out, a vaccine for smallpox. And not only that, during the 20th century, several, antitoxins, several, vaccines, they are found out, some of which I will listed here. All this were able to be found out after the understanding came from, invention of fermentation process, and the finding of the antibiotic from the penicillin. And not only that, recently, during our days, we also emphasize a great, transformation on the entire earth has taken place. That is in 2019, a great beginning of the entire, the world, and its approach has taken place. We see online classes happening now. Earlier, before 2019, we don't know what is an online class, but after 2019, we know what is online class, how to attend an online class. And we all, know at least one from our family, they work from home. Even today, they are not, not at all going to office. They sit at home, do the work. And even today, now, we also attend virtual conference, FDPs, and several other things, sitting from, home. We attend, several lectures across the globe, all because of one, microbe. What is that? It is the COVID-19 virus, which marked a great beginning, not only academically, it changed the entire face of the earth. We have, so many changes happening on this earth. Even today, the e-learning platforms are developing. Not only that, people are used to do, transactions through online. So all these are happening because of one virus. Although earlier, so many institutions were pulling, the students and the teachers to go for online. the central government is pushing for online transfer of money. All those were done. We were not able to move inside, but then this one virus has changed everything on this earth. Not only not only in European countries or American countries, even through the different ends of earth, they started using online platform for various purposes. And today we are also online, sitting at home, traveling in our buses or car. We are listening to the lecture. It is because of the virus. And I think we should thank the virus for making us understand what online is, and making us to make use of the online facility we have today. And I think you all will join with me, agree what had happened. We have seen in our eyes. So although I was telling several things, about the microbes which has done, microbes that has done in the past, and we have also seen during our days, the entire globe is changing for a different environment, and we call it as new normal. Okay, this is the new normal. We operate both mode, virtual mode, and offline mode. So that is how the world has changed now. And not only that, there are several things which happens on this earth even today, and there are so many questions we ask nowadays. What can be done? And I tell you, there are few things which we need to address nowadays, because we can see the climate is changing, the global warming is increasing. For all this, the answer is the microbes. Okay, microbes can absorb and release carbon on this earth. Okay, the Japan is coming out with a a good carbon absorbing ideas. That is, they are going to keep the algae in roadsides, where that can absorb the carbon on the roads. So they are coming up with ideas. We have environmental pollution, such as waste release. So for degradation of the waste, I think the microbes, and degradation of water pollution, the microbes are the answer. We all know several examples. The algae, which plays very important role in wastewater treatment, that could be a very good answer for environmental wastewater. And alternate for fuels, the microbes are the answer. And for treatment of the cancer, the microbes are the answer. And whatnot, if you ask any question, I think the microbes has answers for everything. And if we can rely upon microbes than any other organism, I tell you, I think they have the good answer. But it is we have to think on this line to answer all the questions in the future as God. And with this background, we also do a few things in our lab, thinking the global problem which we are going to face in a near future. One such global problem we are going to face in the recent future is food problem we are going to face. The agricultural lands are dwindling. The cities are expanding due to developmental activities, and therefore the lands are acquired. The farmlands, especially, they are acquired in for road laying or the expansion of the city, whatever may be. The lands are acquired, and therefore there is a land problem. And another problem which are going to come is in 2040. India is going to face a great water scarcity. So water scarcity is there, land problem is there, and then the food problem. You would have seen during September month, Zimbabwe government has issued an order to kill 200 elephants in order to feed their own people. If this is going to be the scenario, there is, a drought in, African countries, Namibia, Zimbabwe. So if this is going to be the scenario, and we have also seen, few months back during May, Bangalore, suffered a lot without water. And last year there was, a news in about Ooty. There is no water in Ooty. So all this is happening around us. If this scenario is going to continue, I think the world is going to be in trouble for production of food. And if there is going to be a trouble for production of food, and we'll have to look for alternate food that can meet the food problem, and that too in terms of proteins. And keeping all this in mind, we have come up from our laboratory to find out few species that are the alternative that can be generated as a food easily. And one such food is mushrooms. And here during our search for mushroom, we have identified a species, Laetiforus sulphureus, from South Indian forest. And we have raised the culture, and this is a culture we have raised. And using this culture, we have started cultivating that. And before that, we have worked out what is the best, why should we go for cultivation. And you can see the protein which is there in this mushroom is 35 percentage is protein, and 48% is the carbohydrate, and the others are less, and the fat is very less. And with this background, we have also compared this species with the other reported species about the minerals. You can see all the other mushrooms and this mushroom which has very good mineral content. Okay, so with this mineral content and the protein content in this mushroom, we thought this could be a good candidate in order to meet the global food demand. There are so many other mushrooms which are there cultivated, and this can also be one such mushroom in the race, because you can see this mushroom has very good shelf life. This will not decay even if you don't keep it in fridge. If you keep it outside, this can stay back for more than 5 days without refrigerating. And if it is going to be refrigerated, that can be kept for more than a week, at least 15 days we can keep it. And the shelf life is more, and therefore we thought this could be a good candidate, and we started cultivating this. And we have also estimated the vitamin content, and we found that has a good vitamin C source. And therefore, with this an idea, we started using that mushroom for cultivation, and we are still on the race. And another one, a good candidate for cultivation is the polyporous species we collected from the South India. This is again a very good mushroom where the shelf life is more, but grows into a bigger size but thin in nature, whereas the previous one is fleshy. This one is very, very thin and has a different texture when compared with the other one, but the shelf life is more. And we have worked on the spawn preparation for this mushroom. And this one is not being known as a edible one in India, whereas in Malaysia this is a very good edible mushroom. And the tribals there, they cultivate this mushroom, and then they consume this. And therefore, as it is not known in India, we started cultivating, and we have standardized the protocol for spawn preparation. This is already published one. And we have also produced the bed where we performed the experiment with different substratum, and we found again this mushroom can adapt to any condition, but then it grows very well in wood chips along with the paddy straw, where it flourishes well. And we have also done the harvesting, and we have also extracted one good pigment from this mushroom. And this is the pigment we have also characterized the pigment. and we have done the characterization, and we have also used up in our foods, and we have also tested the toxicity and found it is not a toxic one, and it is also having anti-cancerous activity, and that I'm not presenting it. Another wonderful mushroom which we worked on is a anti-cancer mushroom. This is in general they are called as chaga mushroom. There are few chaga mushroom which are well known, and it is worked in the Chinese system of medicine, whereas few species which are occurring in South India has not been worked out, and we during the biodiversity studies we collected such species, and few species we have worked with this Felinus nilgiriensis, Felinus sublintius, and Felinus mimosus. This is the morphology of this species, and we have isolated a compound from this mushroom, and we have characterized this, and this is also published, and this is the crystal we have obtained from this mushroom, and that possesses a very good antioxidant activity. And this is the medicinal area which where that can be a solution for cancerous uh, treatment. And we still work on the cell line studies that we did not complete but the other things are published already. And another area we the fungi can occupy and give solution is due to uh, usage of the fertilizer, chemical fertilizer, our land health is suffering now, and it is high time that we have to revert the land in a good condition. The microbes, soil inhabiting microbes has been badly damaged due to usage of chemical fertilizer, and the pesticides we are using. The water has got contaminated, and because of which uh, because of which the bio accumulation and bio magnification, all sort of environmental problem is coming up, and it is high time we think about solving all those problems. And one such area we have also concentrated is a natural fertilizer using a microbe, and we have used the AM fungi, and we have isolated few species from different soil and different places, from from seashore area to mountainous area. We have collected sample, and we were able to isolate few species, and these are the Acylospora, Glomus, and Gigaspora are the common species which we have collected from many places. And then these are the different trees which has association with this uh, uh, mycorrhizas. And after that we have employed in different crops, and one crop I've just shown it here. And it shows a remarkable difference in the improvement of the soil content. This was the experiment we have conducted. We can see the remarkable change even within 1 month. We can see an increase in the nitrogen content, potassium content, and the phosphorus content. So this is within a month. So if this is going to be employed continuously in a particular soil, probably the soil health is going to be reverted back into the original condition as we had at least 20 years back. And the soil microbes are going to revive once again. They are going to provide health to the plant, all those event. And soil biodiversity is going to be reverted back. All this can happen when we can employ this natural fertilizer instead of using the chemical fertilizer. But coming back to this takes time, and it is our duty, the duty of the biologists, to bring awareness to the farmer community to make use of this fertilizer. And we are in in the way to produce fertilizer using single species. But now we have used only with the mixed species. It is better to have single species working with a single plant. That could be a good idea in the future, and we are working on it. And this entire earth belongs to the microbes. We just live here in this world of microbes. And when we think that the world belongs to us, then we start exploiting the earth for ourselves. But it is not like that. The world belongs to microbes. They change according to what they wanted, as we have seen a few examples earlier. So they are going to continuously keep on changing. Even today the microbes are working on this earth to change it into a desired condition and desired condition. And we just live on this earth. And if we can live in harmony with this microbes and the other life forms on this earth, and surely I think we can do a marvelous thing together with the microbes. So only thing is we need to understand microbes can do anything on this earth. Okay, this is one such uh, change the microbes has done in the for the New Zealand. You can see there is a logo here in the tail portion. So here also there is a tail, there is a logo here. Okay, this logo is not just a simple design. It is a design of it is a design or a logo which has been mimicked from a mushroom. So here it is the Mycena which formed the logo of the New Zealand Airlines. So this is the Mycena species, which looks similar to it, and that is taken from by the New Zealand Airlines, because of the remarkable contribution of or in the biodiversity of New Zealand by this particular mushroom. And yeah, with this small note, I just want to thank everyone for your patient listening, and also the organizers for giving me the opportunity to present a few things about the microbes. And I am happy to take up a few questions.
Thank you, sir.
Yes, sir. Yes, sir.
Thank you, sir, for your detailed presentation, and your expertise has been enriched to our understanding and broadened it to our horizons. It's time for query section. If there are any questions, kindly post the questions in the chat box.
That's not
Sir, some of
already
the participants are asking. Yes, sir, yes, sir. Some of them are already asking. Can you explain about trap soil?
more,
Sir?
sir.
Yeah, I'm my Zoom is not there. I'm just coming. Yeah, the trap soil is an experiment where we bring the soil from the field, and we do sieve. The sieves is of different sizes, and after doing the sieve and getting the spores, we do trap them in the sterilized soil, and we observe under microscope, and then we select particular spores, and then we trap them in the soil and use it for the culture. so which is called as the trap soil, which we do.
Okay, sir. Thank you. So another one question is there, sir. If nitrogen and phosphorus, potassium of trap soil increase in a month, if it obviously has a relation with pH and electrical conductivity.
Pardon?
Sir, can I repeat, sir?
Yeah.
If nitrogen, phosphorus, potassium of trap soil increase in a month, it absolutely have a relation with pH and electrical conductivity.
Yeah, probably. I think there is a connection between all this pH and the electrical conductivity. There is no doubt in it.
Another one question is what culture media you have used to lattice isolation, sir?
It is the simple PDM medium for just isolation. And if you want to do other work, you can choose a different medium. But for isolation, I think potato dextrose agar medium is enough.
Okay, so thank you, sir. Thank you so much for your knowledgeable response.
Rashma ma'am?
Yes, sir.
Okay, I think there is another
Another
question.
one.
You can just go ahead.
Yes,
Yeah.
another one question. I'm a researcher in soil science. Which method you use microbes isolation in lab?
Serial dilution method for isolation of the soil microbes.
Rashma ma'am, I think there are no
Yes,
other questions.
no question is there, sir. Yes, sir.
Okay.
Thank you,
Kumar
sir.
sir,
Thank you so much.
one second, sir. I have some 2 or 3 questions to ask to him.
Yes, sir.
Kumar sir, can you
Sir,
able to hear me?
I'm here.
Thank you for your wonderful insight. Certainly, all the participants as well as we all just agree with you that the microbes have changed the world. Okay. So just a simple question to you. As a renowned mushroom taxonomist, so when you go to the tropical forest for field visits and other things for mushroom cultivation, what is the main thing you notice? Okay, so nowadays we can able to see a drastic climate change due to global warming. We get a lot of rain. Okay, so what is the general thing you just observe whether is there any change in the mushroom species or the what to say vanished common species, or you can able to notice any change in its morphology or something? Can you just
Yeah.
provide some insight?
Yeah, due to climate change, surely there is a change in the biodiversity pattern, which can be emphasized from the new species which are being discovered from the tropical regions. And new species are being discovered because one thing is it is continuously evolved. That is one reason. When climate is going to change, maybe a 1 degree change is also a remarkable change for a microbe to evolve and to be revealed. And the world is trying to stop IPCC. They have come out with a resolution that they have to restrict a temperature rise of 1.5 degrees until 2030. So if it is now a present condition, if it is growing like this, that will cross 1.5 degree change within 2030. But the steps are taken to restrict within 1.5. When that 1.5 is going to be restricted, even in that condition itself, there is going to be a new species evolving from different regions across the world. And morphologically, at present we don't see much changes in the species which we are taking. But then new species are being evolved, which may be due to not, I'm not saying this is due to, maybe due to the climate change.
Thank you, sir. Thank you for your answer. Just another
Thank you.
Thank you,
question. Just give some insights on lichens. Okay, so nowadays we can't able to see so much of lichens in trees. Is there any what to say alarming situation for the human being or to the environment?
Yeah. Lichens are very, very sensitive. They require climate, and the environment should not be polluted, as we all know. Wherever the pollution is very, very less or minimum, I think you can see three types of lichen: the crustose lichen, foliose lichen, and fruticose lichen. When the environment is polluted to an extent, then we can see a crustose lichen and foliose lichen. When the pollution is more, we can see only crustose lichen. If the pollution is exceeding particular limit, and you cannot see lichen at all. Probably in cities, you cannot see any lichen. But when you visit our campus, Madras Christian College campus, which is a forest campus, which is of 350 acres, where we are able to see the foliose lichen as well as crustose lichen, which means inside the campus, though it is in city, inside the campus, I think the pollution is very, very less. We are fortunate to see both the lichen, that too in the deep forest, not in the place where it is closer to the roadside. When we go deep into the forest, I think we can see two different types of lichen. And probably due to tourism activities and other activities, even in the hill station, probably we may lose few diversity. And we'll have to think about the development of the tourism and lichen in all this aspect.
Thank you, sir. Thank you for your clear explanation as well as your wonderful insight through this FDP. Thank you. Thank you very much for spending your valuable time and accepting our invitation. Thank you, sir. Thank you very much.
Thank you so much, sir, for having invited me for presenting this. And I thank the institution, Sacred Heart College, for having given me this opportunity. Thank you, all the participants.
Thank you, sir. Thank you, sir. Sir, one more question is there, sir, from participants. If there are any demerits for isolation for anti-cancer mushroom in laboratory?
I think isolation, there is no demerits. I don't know whether your question is that or something. Isolation, there is no demerits.
Okay, sir. Thank you, sir. Thank you, sir. Once again, thank you so much, sir, for your knowledgeable response. Dear participants, feedback link is posted in chat box. Kindly fill it as soon as possible since it will be activated for few hours. Thank you once again for making this FDP a resounding success. Thank you all. Thank you so much, sir. And
Thank you.
participants, we are kindly requested to stay connected for tomorrow. Yes, sir.
I think this question is not addressed there. Dr. Kumar, I'm researcher in soil science. Which method you use microbe isolation in laboratory?
We only use serial dilution, and for here for mycorrhiza we have used the sieving method to isolate the WAM fungi. So in general, if you ask, it is serial dilution, but we have used the sieve method to isolate the spores of the AM fungi.
Okay, sir. Thank you. It was a question of Ms. Selva Lakshmi. Thank you. Thank you, sir. Thanks for your
Thank you, Nia, sir.
Thank you, sir.
Thank you, sir. Thank you very much. Have a
Thank you,
nice
sir.
Thank you. Thank you, sir. Thanks for your availability.
Thank you, Nia, sir.
Thank you, sir.
Ms. Reshma, can I
Yes,
wind up,
sir.
ma?
Yes, sir. Yes, sir. Participants are kindly requested to stay connected for tomorrow's section at the same time. Thank you, dear participant, and thank you, dear source person. Thank you, sir.
Thank you, madam.
Thank you.
Thank you, Dr. Kumar. Thank you, all of you. Thank you.